Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we started discussing immunology recently and we have been discussing about the cells of immune system. So we started with the discussion of immunity, the types of immune system that we possess, the innate immunity as well as the adaptive immunity, right? Then we discussed about the cells of immune system. We started discussing about hematopoiesis, the phenomena of generation or maturation of blood cells from the hematopoietic stem cells. So we discussed about each of the different lineages that will be formed from the hematopoietic stem cells. So we discussed about each type of different cells that are the part of our immune system. Either they could evoke the innate immune response or they could evoke the adaptive immune response. So in the last class, we learned about natural killer cells, and in today's class, we are going to learn about lymphocytes, the B lymphocytes and T lymphocytes. So in today's lecture, we are only going to talk about the basic stuff about those B cells and T cells, the surface markers, what are the roles of those cells, and what are the organs of origin for those cells. But the development of those cells, the activation, maturation, all of those stuff that will be discussed in the adaptive immunity lectures. So today we are only going to talk about the cells as in general, the surface markers that will be expressed by those lymphocytes, and that's it. So let's get started with today's lecture. So the hematopoietic stem cells, okay, uh, I've been discussing this from past few lectures, that it gives rise to the myeloid lineage as well as the lymphoid lineage. So hematopoietic stem cells, they could get differentiated into common myeloid progenitor or a common lymphoid progenitor. So this common lymphoid progenitor is going to give rise to the cells which do not have granules in their cytoplasm. So they are agranular and they are referred as lymphocytes. But there exists a category of the granular lymphocytes which we discussed in the last class, and that is natural killer cells. So this common lymphoid progenitor, it is going to give rise to the B cells, the B lymphocytes. It is going to give rise to the T lymphocytes, means the T cells, and it also gives rise to the natural killer cells, right? Where the B cells and T cells, they are agranular in nature, whereas the natural killer cells, they are granular in nature, right? So when we were discussing about hematopoiesis, we learned about histopath, the reagent which is helpful for differentiating those cells, for separating all of those cells in the pool of a blood. So from using histopath, we can separate the PBMCs, okay, which majorly consist of lymphocytes, and they also have monocytes in them, right? The major part of PBMCs, peripheral blood cells, they are the lymphocytes. So it has B cells as well as T cells. So for the identification of B cells and T cells, for distinguishing them apart from the natural killer cells, we can use the morphological features because these are agranulocytes, whereas the natural killer cells, they are granular in nature. That is why they are also known as granular lymphocytes, right? So we can differentiate both of them based upon the morphology. But for distinguishing B cells and T cells, for separating B cells apart from the T cells, we need to have other way to process this information, process the identification, because B cells and T cells, they do not really have much differences morphologically, so we cannot really differentiate them when we view them under the microscope. And for that matter of purpose, we use the cell surface proteins, the complex of proteins that is present over the surface of those immune cells, and these are known as cluster of differentiation. So the molecule, okay, so what is cluster of differentiation? So cluster of differentiation is it is the cluster or the category of the cell surface proteins, which could act as the signaling protein, which could act as a simple cell surface receptor, or they can also be the adhesion molecules. So they form a cluster over the surface of those immune cells, and with the help of identification of those cluster of differentiation, with the help of identification of those cluster, we can identify those cells, because all of those cells, they are going to have different clusters of those molecules at the different developmental stages, right? So we have been discussing about CD molecules. These are abbreviated as CD. So we have discussed about CD3, which are present on T cells. We have discussed about CD34, which are present on the hematopoietic stem cells. We also discussed about CD16. We discussed about CD56 and many more. So all of them, they are different clusters of the receptors or the markers which are present over the surface of that cells. So the cluster of differentiation, this is a way or a protocol to differentiate all those those surface proteins in a cluster, and to be using them we can identify those particular immune cells. So these are the cell surface molecules which enables us to identify and classify immune cells. So these are the cell surface molecules which enables us to identify and classify different types of immune cells. Immune cells. Right? So using those, we can differentiate between those immune cells. So those molecules, they are cell surface proteins, and they just not differ in the different types of cell, but they can also differ in the same cell if the cell is at the different developmental stages, right? So the hematopoietic stem cells, which is going to give rise to the different progenitor cells, this the CD molecules that will be expressed by the hematopoietic stem cells, which is CD34, right? That will be different from what the cells, the progenitor cells are going to express. So basically, it is the more differentiated cell, and it is even if the origin of that particular progenitor cell is same, which is hematopoietic stem cell, even though the origin is same, both of them, they are expressing different CD molecules, right? So this is how the CD molecules, they are different, not only upon the different functionally different cells, but even the same cell will express different surface proteins during the process of development. So the cluster of differentiation will differ in functionally different cells, but it will also differ for a same cell at different developmental stages. Right? So this is how cluster of differentiation helps us to differentiate those immune cells, as well as to identify which type of stage that cell is right now in, right? So which developmental stage that particular cell is, that will also be identified using cluster differentiation. So for that purpose, we use a set of monoclonal antibodies which is going to specifically bind to those surface proteins, and this whole protocol of identifying those different classes of the cell surface proteins is known as cluster of differentiation. So the nomenclature of cluster of differentiation is according to the order by which they are found. So the CD3, this particular this particular cluster was found before CD4, and that is why it was named as CD3, and this was named as CD4. Whereas the CD19 or CD16, they are found much later. So the nomenclature is quite simple for the cluster of differentiation. We simply name them according to their date of the discovery. Okay, so the first discovered one will have a lower number, and the later discovered one will have a larger number. So I have been discussing about those different types of CD molecules that are present over the different cells as their surface markers, but I did not really tell you what that is. So I hope the cluster of differentiation, what does that mean, how this protocol works, is clear to you all. So let's now start discussing about lymphocytes, the B lymphocytes and the T lymphocytes. Okay, so as been previously discussed, the B lymphocytes, they are a part of adaptive immunity. Means they are going to express the antigen specific receptors over their surfaces. So what does B lymphocyte do? They recognize antigen, the specific antigen which is present over the pathogen, or simply the antigen has been entered in the body. So they recognize that particular antigen and they produce antibodies against those antigens. Okay, so the antibodies which will be produced by the B lymphocytes or the B cells, they are the part of humoral adaptive immunity. So these antibodies, they are actually immunoglobulins, and those immunoglobulins are the highly glycosylated molecules. So these are actually the glycoproteins. I will be writing immunoglobulins as Igs. So these antibodies, they are a part of humoral adaptive immunity, which is produced by the B cells, specifically the plasma cells. Okay, so these B cells, they also act as antigen presenting cells. Okay, this has been discussed previously, that the dendritic cells as well as the macrophages, and alongside them B cells, they also act as the antigen presenting cells. So they are going to present the antigens with the help of MHC molecules to the T cells, and that will result into the activation of T cells and initiation of immune responses by those particular T cells. So this is how they B lymphocyte, they just not help into the humoral immunity, but they are also part of the cell mediated they are also conducting the part in their cell mediated immunity by activating the T cells. So they also does the job of antigen presentation, and besides antigen presentation, they produce cytokines. Okay, and the production of cytokines by the B lymphocytes, that will be help in shaping the innate immune response, that will induce the innate immune response. So by now you must have got an idea that all of the immune responses, whether it be an innate immune response or whether it be adaptive immune response, all of them are interlinked. Okay, so many cells are going to help into those interlinking phenomena. So this is how the B lymphocyte, which are actually part of the adaptive immunity, they produce cytokines to shape the innate immune responses. So this B lymphocytes, this prenatally before birth, they are found in the fetal liver. So the prenatally, okay, so prenatally B cells, they are found in the fetal liver. So fetal liver it has the job of development of B cells before birth, but after birth, those B cells, they develop from the hematopoietic stem cells in bone marrow. Right? The B cells, they were first discovered by a researcher known as Max Cooper. Okay, so they were discovered in 1960s by Max Cooper. So Max Cooper, when he was, when he started studying, studying the immunology, what he did, Max Cooper, he showed that the production of antibodies, it was completely abrogated in chickens after the surgical removal of bursa fabricius. So bursa fabricius is a specialized organ in the chickens, in the birds. Okay, so let me write it down here for you. Bursa of fabricius. This particular organ is found in the birds. So when in the chicken, this bursa of Fabricius was removed, and then those chickens were irradiated. So at that point of time, Max Cooper found that after the surgical removal of this organ and the irradiation of the chickens, he found out that there was complete abrogation of the formation of the B cells. This means that this particular organ plays an important role for B cell development in the birds, and that is why those particular cells, they were named as B cells due to the organ of origin. So Bursa of Fabricius has the job, it is the primary site for the development of the B cells, okay, in birds, okay, but in humans, in the mammals, it is bone marrow. So this is how the B cells got their name after the organ of origin, Bursa of Fabricius. So those B cells, they when they do the antigen presentation, so they are going to activate the CD4 positive T cells via MHC class 2 molecules, right? We have discussed this previously as well. So they are only going to activate the CD4 positive T cell, not the CD8 8 T cell. So they are not going to activate the cytotoxic T cells. They're only going to activate the T helper cells. Okay, so this is a very important point that you need to note. So the surface markers of the B cells, means the proteins, the clusters that are found over the B cells, they are as follows. So let's list down the surface markers of the B cells. So there are different surface markers present, such as CD19 and CD20. Okay, so CD19 and CD20, both of them, they are the markers for B cell lineage. So we can identify the B cell lineage with the help of those markers, CD19 and CD20. Then besides those markers, there exists CD21, which is actually a complement receptor. We haven't discussed about complement system yet, so don't worry about this. For now, just know that this is a complement receptor. Then there is the CD40, which interacts with the CD40 ligand for the T cell activation over the T cell. So this CD40, we can say that this is a way to to have an interaction between B cells and T cells, right? So B cells have the CD40, okay, they have the CD40, and T cells, they have CD40 ligand over their surface, and this is how the interaction will be seen between the B cells and T cells, and that is going to activate the T cells. So these are the T cell activatory receptors that are present over the B cell. So CD40 binds to CD40 ligand over T cell, and it does the activation of T cell. Okay, so besides those markers, there are present the B cell receptors, BCR, B cell receptors. They are also present over the B cell surface. So basically, these are membrane. These are membrane bound immunoglobulins. So they are going to work as a receptor for antigens. So these are the antigen receptors. Okay, so they are called B cell receptors because, well, they are the receptors for the antigens which are present on the B cells. So they are also going to activate B cells. So once the particular antigen goes and binds to B cell receptor, that is going to result into activation of BCR, and activation of BCR will result into the activation of B cells. Beside those surface markers, there are the MHC class 2 molecules present over the B cells, because, well, B cells are the antigen presenting cells I mean, one of the antigen presenting cells, and we have discussed this that the antigen presenting cells have MHC class 2 molecules. Well, MHC class 1 molecules are there on all types of cell of an individual except the ones which lack nucleus. So only RBCs are the ones which does not possess MHC class 1 molecules. Rest of the cells of the body, they do have MHC class 1 molecule. Whereas MHC class 2 molecules, they are only present over the antigen presenting cells. So the MHC class 2 molecules that are present over the B cells, they are going to result into antigen presentation, okay, to the CD4 positive T cells, which are also T helper cells. Okay. Besides those, there are CD18 and CD86. These are actually the co-stimulatory molecules for T cell activation. So this is the one of the molecules which is going to mediate the T cell activation. And for this activation, there are present some co-stimulatory molecules. Means these are also supporting the activation. Okay. So this co-stimulatory molecules support the T cell activation. Right, so these are the cell surface markers for the B cell, and they have their own different roles. So what are the different jobs that the B cell is going to do? So the B cell, it has different roles to perform inside the body. So it is, first of all, it is mediating the antibody, it is mediating the adaptive immune response by the antibody production. So the very first and important job of the B cell is antibody productions. It also, does the antigen presentation. It also secretes cytokines. Okay, so the cytokines which are secreted by B cells are the different cytokines such as IL-10, right, IL-6, and GM-CSF. The granulocyte macrophage colony stimulating factor, which is going to increase the efficiency of macrophages and the granulocyte. So they are going to increase the engulfment capacity of those particular cells. Right, so this is also a cytokine signaling molecule. And besides those jobs, the other jobs of the B cells are the immune regulation. Okay, so they are also modulate the immune responses, and besides those, they also has a role in memory generation, the immunological memory generation. We will be discussing about this particular part in the B cell lectures of the adaptive immunity. Okay, so when we will start discussing about adaptive immunity, we'll discuss about those particular roles, right? So let's now discuss about T lymphocytes. So we have done this with the discussion of B cell. Let's now discuss about T lymphocytes. So the T lymphocytes, they were named as T cells, T lymphocytes, because these are actually matured, their maturation is seen in thymus, and that is why they were named as T lymphocytes. So here I'm not going to discuss too much about T lymphocytes. There are just, I'll only be discussing the types of the T lymphocytes. So the T lymphocytes have two types that we have been discussing since a few lectures. The cytotoxic T cells, which are also known as CD8 positive T cells, and there are the T helper cells, which are also known as CD4 positive T cells. So there are different types of T helper cells that are going to have their different roles. Some of them, they are the activatory T cells, while other of them are the inhibitory T cells. For example, there is TH1. So this TH1, this is a T helper cell type. It is important for intracellular pathogen elimination. So this plays a role in intracellular pathogen elimination. Then there is TH2 cells. Okay, so those cells, they have their role in inducing strong antibody responses. So they are going to mediate the B cell mediated immunity. So they are going to induce strong antibody responses. This particular T helper cells, they are going to help B cells to generate the strong antibody responses, and they are also, they're also a part of allergic responses. So they are also responsible for generating allergic responses. So besides those T helper cells, there exist the Th17 cells, Th22 cells, okay, and TFSH cells, okay. So they also have different roles, such as the T17 cell. This promotes inflammation, okay, and this also promotes the autoimmune diseases. Okay, when the tolerance property of the immunity is compromised, then the individual suffers the autoimmune disease, and that is mediated by the Th17 cells. Then the Th22 cells, they are a part of mucosal immune response. Okay, in the mucosal region, the food is also the foreign substance. So why doesn't food it induces immune responses? Because at that particular site, the tolerance capacity plays a very important role. So there is a perfect balance, and whenever this balance is disrupted, there is going to be inflammation, and there are going to be some immune responses which are referred as mucosal immune responses, right? So the tolerance capacity of the immunity plays a very important role in those particular areas, because food is very important for us to survive, and it shouldn't generate any immune response. But many times the food that we are consuming, it has pathogens. So all of those things, they are kept in check by the immune system, and if something goes wrong, then the mucosal immune responses, they are generated, and this Th22 cells, they also show inflammatory responses. Okay. Then there are TFH cells, which are actually mediating the B cell immunity. So these are the antigen, these are going to mediate antigen specific B cell immunity. Okay. So all of those T helper cells that I discussed here, all of them, they are activatory. But as we need an accelerator, we also have a need of a brake. So there is inhibitory type of the inner T cell, which is going to regulate the whole T cell responses, which is known as Treg. So these are the regulatory T cells, which basically dampen the immune response. So they are going to, inhibit the T cell activation. They are going to totally dampen immune response. So they are basically the brakes over our immune responses. And these are very crucial so that the inflammation doesn't go overboard and we can keep the immune system in check. So these are the different types of T helper cells that are found in our body. So in the last class, I told you that the natural killer cells that we discussed, which are also part of lymphoid lineage, but basically they are the innate cells, right? So there is a subtype of there is a subtype which resembles to both natural killer cells and T cells, which is referred as NKT cells, right? So let's discuss about this particular subtype. So those NKT cells, they resemble to both NK cells and T cells, and that is why their name is NKT, right? And those are also referred as innate like lymphocytes, because natural killer cells, they are the lymphocytes which are innate, which are a part of innate immune system. But these NKT cells, they are not, we cannot really categorize them into innate immunity or adaptive immunity, because this is a subtype which is going to resemble to both of those cells. So they have the cell surface receptors for the NK cells as well as for the T cells. So whenever in the population of the immune cells, let's say these are few cells, okay, and you really don't know which type of immune cells those are. So then you go for the cluster of differentiation, the CD molecules that are present over the immune cells. So a few cells have those CD molecules, right? They are, they are expressing those particular CD molecules, and few other cells they are expressing those molecules. Okay, but then there are a few cells in this particular population which has the expression of both of those markers, and this particular subtype is known as NKT cells. These red ones are, let's assume that these are T cells, so they are expressing T cells markers, and these are the NK cells. The pink ones are NK cells because they are expressing NK cell surface markers. So the cells which has the expression of both NK cell surface marker as well as T cell surface marker, these are referred as NKT cells, natural killer T cells. Okay, so those natural killer T cells, they does not only have the surface marker expression for both of the cells, but they also resemble to both of the cells in the killing mechanisms. So they are going to do the jobs similar as NK cells as well as T cells. So these particular NK cells, they have cell surface markers like NK cells. For example, CD56 and NK 1.1. Well, this particular marker is found in mice, not in humans. And then there is NKG2D. So these are the NK cell markers that we discussed in yesterday's class. So this will be expressed by NK T cells. So these natural killer T cells, they will express those markers, and besides those markers, they are going to express the conventional T cell receptors, the TCRs. Okay, so they will express both of those receptors. But what makes them different from those both cells, from NK cells as well as T cells? the the point that this differentiates them from the T cells, even though they have T cell receptors, the conventional T cell receptors, this particular cells, they are not MHC restricted. Okay, so they are just like NK cells, which are not MHC restricted. Means for the killing of the pathogen, they do not need the MHC molecule help, and for antigen presentation, they do not need the help of MHC molecule. So they resemble to NK cells in a way that they can recognize the antigen directly, and they resemble to the T cell in a way that they are not MHC restricted. They do not need MHC molecule for antigen presentation, but they do need antigen recognition. Sorry, but they do need a molecule known as CD1D, which is resembling to MHC1 or MHC class 2 molecules, but which is not a part of those molecules. So these are not MHC1 or 2. These are different molecules, but these are also specific molecules CD1D, which are required by NKT cells for the for antigen recognition. So how does this NKT cell resemble to both of the cells? It resembles to the natural killer cells because they can natural killer cell, they can directly recognize the antigens. In the same way, those NKT cells, they can also recognize the antigens, but the recognition of the antigens which are recognized by natural killer T cells, they are not conventional. So this particular cells, they recognize non-conventional pathogenic antigens such as the lipid antigens or glycosylated or the glycolipid antigens. Okay, there are bacteria, there are certain pathogens which have, instead of proteins as antigens, they have lipids and glycolipids. Okay, so any foreign substance that will be going to, identified by our immune system, that is referred as antigen. I have discussed this previously. And when that particular antigen induces the immune response, it is referred as immunogen. So the conventional antigens are proteins. Well, because the proteins are present abundantly over the cell surface, over the surface of pathogens. Whereas the non-conventional ones are the lipids and the glycolipids. And a very nice example of this is the Mycobacterium tuberculosis, which Mycobacterium tuberculosis. This particular bacteria has those non-conventional antigens. So the non-conventional antigens, they are going to be recognized by those NKT cells, and they can directly initiate their response. and this is how they resemble to the NK cells, and they also resemble to T cell receptor because the recognition of those non-conventional pathogenic antigens that will be done by the CD1D molecules. Okay, so the recognition of those particular non-conventional antigens will be done by CD1D molecules, which does not belong to either MHC1 or MHC2 class of the molecules. Okay, so this is how the natural killer T cells, they are a subset of the T lymphocytes, which resembles to both natural killer cells or the natural killer T cells. So we can say then that they are a subtype of natural killer cell, or they could be a subtype of the T cells. Okay. So I did not discuss the T cell markers. Let's just list down the T cell surface marker here. So there are few, surface proteins that are present over the T cell. So these are, first of all, I have been discussing throughout many lectures, that is CD3, which is present over both T helper cell as well as the cytotoxic T cell. Then there are CD4 and CD8, which are going to differentiate into those T cells. So they are going to have by the expression of CD4 and CD8, the T cells will be subgrouped into two different categories, the cytotoxic ones and the helper ones, right? Then they can express they can express the T cell receptor, the conventional T cell receptor, which are known as TCR, right? Besides them, they also have other CD molecules as their expression, as their surface markers. And with this, I'm done with the cells of the immune system. We have discussed about all of the cell types that are found in our immune system. And in next lecture, I am going to discuss about the other stuff about immune system. I will be starting the introduction about immune organs, the primary lymphoid organs and the secondary lymphoid organs that we have, and then we will discuss all of those organs one by one in detail. So the overview, I'll give you an overview about the immune organs, and then we'll start discussing about them in detail, just like we did for the cells of immune system. So I'll catch you in the next lecture. Till then, see you. Bye bye.